Good evening. Are you able to see the slides? Hello.
So,
Are you able to see the slides?
yes, sir, yes.
Okay. Today we'll be talking about multiple sclerosis, the endophenotypes and the implications. The scheme will introduce the topic and we'll talk briefly about immunopathogenesis and current concepts in pathobiology as we understand today. What is the clinical question which you're going to answer, and what is the exploratory hypothesis? And we look at analysis of flow cytometry and proteomics data. And we learn about UMAPs and heat map, differences between endophenotypes, multifaceted immune dysregulation in MS, machine learning application for the data generated by flow cytometry and the proteomics, and what are the clinical disease trajectories of these various endophenotypes, and implications for research and treatment. And this is, in short, what I'm going to talk in the next 30 minutes. Multiple sclerosis, as you know, is first described in 14th century. In 1868, Charcot published his lecture on sclerosis and plaques. It's a relapsing remitting disorder, affects multiple white matter tracts within the central nervous system, and mostly onset in young adults. There are multifocal areas of CNS myelin destruction. These are called MS plaques. And most important is in multiple sclerosis, the myelin sheath, which is a single cell whose membrane wraps around the axon, is destroyed with inflammation. This is the myelin sheath of a healthy nerve is shown here. And you can see the myelin on light microscopy. Myelin sheaths are around the axons. And there are a couple of oligodendrocytes which are seen here, which is a black arrow which is pointing to one. And this electron microscopic picture of myelin sheath with microtubules and neurofilaments. So this is the nerve conduction, which is in the healthy nerve, as you see in the above. And the down below, when the scarred, the myelin is gone, then the saltatory conduction is affected. So let's look at the immunopathogenesis of MS. MS is the silently it progresses. There's a three-pronged attack. One on blood-brain barrier by the activated immune cells. The blood-brain barrier is breached and damaged, and repeatedly it is breached. The second is there is secretion of inflammatory cytokines which flood the brain, and these are activated immune cells that release the pro-inflammatory cytokines and free radicals. And also the third aspect is the repair is also compromised in multiple sclerosis. The hallmarks are three: inflammation, which can be seen in the histopathology and also on neuroimaging, demyelination, and axonal loss, which is seen as black holes on MRI. So the initial concept of this demyelination is due to a immune dysregulation. The concept was that key role for CD4 and CD8 T cells in this immune dysregulation in MS. Now this concept is being replaced by involvement of other cells like beta cells, B cells, NK cells, and distinct myeloid cells. So it's in fact a multifaceted immune dysregulation. Both innate and adaptive immunity are affected. In this, the innate immunity rapid response is mediated by the myeloid cells, the natural killer cells, and the natural killer T cells also are there. And the slow responsive adaptive immunity is mediated by the B cells, which secrete the antibodies, and the T cells, which are activated CD4 and CD8, they secrete cytokines. So there are drugs which act on the slow adaptive response. So that is alemtuzumab, beta interferon, dimethyl fumarate, fingolimod, glatimarestate, etc. So the innate immune response, the pathogen is attached to the ligand, and the antigen processing cell, which will activate the native T cell, which will give rise to the secretion of interleukins from T helper 1, 2, and 17 cells, various interleukins, these are the cytokines which will cause havoc in the brain. So current pathobiology concepts say that MS is an inflammatory demyelinating disease of the central nervous system. It is hypothesized to be initiated by autoreactive T cells and also other cell subsets, including B cells, NK cells, and myeloid cells, that is macrophages and microglia. The early phase of relapsing remitting MS is characterized by peripheral immune response targeting central nervous system. The latter phase is compartmentalized local inflammation within the CNS and neurodegeneration, which I'll show you in a couple of slides. So you can see the periphery, the cell activation and proliferation of the anterior antigen presenting cell to the T cell, and it secretes the interleukins, and they again activate B cells, those secrete the antibodies. So what result is endothelial cell attachment and migration into they breach the blood-brain barrier and enter the central nervous system and cause the demyelination, and of course, the immune cell reactivation and proliferation inside the brain takes place. That is the compartmentalized one, which I was talking about, the T helper 1 and 2 cells, and subsequently CNS damage and repair takes place. So, and the blood vessel lumen, the lymphocytes, they get attached to the blood-brain barrier, and slowly, they migrate into the brain parenchyma, and the activated cells cause various chemokines, also assisted with microglia and astrocytes, and cause demyelination. So whether evaluating immune signature in early MS subtypes, uh, early MS subjects before initiating DMTs or disease-modifying therapies, will it be helpful in predicting clinical disease trajectory and also dictate the therapy? That's the main clinical question. So to evaluate immune signatures, we have to identify what are those signatures. So this is in fact gives rise to what is called personalized treatment decisions based on pathological principles. So what we do so far is whether one drug fits all approach that we approach, can it be changed to precision medicine in multiple sclerosis? That's the main question which we answer. So the MS, as you know, is characterized by heterogeneity of clinical manifestations. The exploratory hypothesis behind this talk is whether discrete immune abnormalities in blood are surrogates of pathophysiology, and they can be, if identified, do they dictate a certain type of trajectory for the disease and treatment? The immunobiological principles can be used to select a treatment then, or the treatment modality, what can be, whether it is interferons, dimethyl fumarate, or glatiramer acetate in the mild MS. So at present, the treatment selection is based on a combination of clinical and subclinical parameters of disease activity and severity, safety aspects, and patient preferences. But this approach disregards the pathobiology of putative heterogeneity of the underlying immune dysregulation. What is the emerging concept? Personalized treatment selection, precision medicine. So exploratory, this study, which appeared in March 2024, it studied multiple sclerosis endophenotypes, which are identified by high dimensional blood signatures that are associated with distinct disease trajectories. And this study is a landmark study. This happened in Germany, where it's a multicentric cohort study, national MS cohort, 1,200 therapy naive patients less than 2 years from the disease onset, the serum from 309 patients with MS. What they did was the blood samples were subjected to high dimensional characterization of peripheral blood mononuclear cells abnormalities by combining multi-parameter flow cytometry and targeted proteomics. Proteomics, sorry. So, what is the sir? I am in a class, please. So analysis of this flow cytometry and proteomics data, that is the what's called UMAP, that is Uniform Manifold Approximation and Projection Map. It is used for high-dimensional graphic representation of canted and scaled flow cytometry data. So these type of maps I'm going to show you. The flow cytometry data derived from 71 healthy donors and 309 therapy naive patients with MS, and the proteomics data of 40 healthy donors, and 158 patients with MS are studied in detail, and the flow cytometry data in the form of heat map. The heat map is a graphical representation of data where the individual values contained in a matrix are represented as colors. You can see the healthy donors and MS, and the parameters are also scored here. So heat map with hierarchic clustering of normalized flow cytometry parameters. They are organized by groups in this slide. So the comparison of immune signatures between early MS patients and healthy donors is done based on discrete immune cell subsets, cell surface markers, cytokines, chemokines, cytolytic granule content, and lytic activity, that cellular signatures. As you can see, CD8 T cells, CD4 T cells, monocytes, all these things have been studied in comparison with healthy donors. So that they found that there are specific cellular signatures that define mainly three endophenotypes in multiple sclerosis. They are called E1, E2, and E3 endotypes. The automatic grouping of patients with MS by the phenograph algorithm resulted in these three distinct endophenotypes, and they are based on the differential phenotypic data mapped onto the UMAP graph. Then the heat map of the relative frequency of the investigated parameters sorted into 5 parameter groups by phenograph and split by MS endophenotypes. So basically, this jargon, what it tells is there are basically 3 types of endophenotypes which have got a different different immunological signatures based on the expression of cellular parameters, and there are differences in these endophenotypes between immune cell subsets, cell surface markers, cytokines, chemokines, cytolytic granule content, etc. So there are differences between. So how they are going to translate onto the clinical manifestation? That's the main important thing. So what are these differences and how the differences are identified? You can see in these graphs here, the healthy donors are compared to E1 endophenotype, E2, and E3 phenotype. These endophenotypes of early MS patients are classified dependent on the 10% most significantly altered cellular parameters. So these cellular parameters have been studied by flow cytometry, and the graphs have been constructed for E1, E2, and E3 when compared to healthy donors. The HD is healthy donors. So what they did was these cellular signature parameters and immune signature parameters, they have they have been they are used to define these endophenotypes again further, and the multifaceted immune dysregulation has come into vogue. So the initial concept, as I earlier mentioned, gave a key role for CD4, CD8 T cells in this immune dysregulation. But this is replaced by the notion that there are involvements of other cells like B cells, NK cells, and distinct myeloid cells. In E1 endophenotype, the percentage change in CD4 T cells, the increased T cell expression of Th17 subtype, interleukin 22, and GM-CSF, and E3 subtype increased CD8 T cell compartment, E2 changes in NK cell. All these things say that when the disease starts, it can have a prototype neuroinflammation type or a prototype cell damage or neuronal damage. And what is guiding these endophenotypes is a HLA subtype, which determines the respective endotypes. So the endophenotypes on HLA, these cellular immunological signatures are correlated with distinct pattern of HLA subtypes. For example, E1 is correlated to HLA DRB 16:1. E2, there is increased frequency of HLA 11:1, HLA B14:02, HLA C08:02. E3 is decreased frequency of HLA E29:02. So distinct immunological subtypes in MS are shaped by MLA background, HLA background. So then comes all this data is fed into a machine learning algorithm and a machine learning model And this longitudinal cohort, which they have described as a derivation cohort of 3809 patients whose blood samples were collected. And once the model is developed, then uh, they have validated in another cohort of 232 patients whose the data is unknown to them. So what they used a high dimensional flow cytometry and serum proteomics data. So as I said, there are three distinct endophenotypes mediating inflammation and early structural damage. So endophenotypes could be classified with 95% accuracy using a combination of 23 cellular and 3 soluble protein parameters, which were brought out by the flow cytometry and proteomics. See, the discovery cohort or derivation cohort, which is a machine learning model, it is trained with this data from 309 patients with early MS using these cellular and soluble specific parameters. And after the model is trained, it is validated using another 200 odd patients to predict different endophenotypes from the unseen data. So these are the validation cohort, and this is the derivation cohort. You can see the various colors. As I said, there are phenograph, heat map, and U map. These are the terminologies which are very well known to genetic scientists and immunologists. But we are not aware. But a suffice is to know that there are differences between the serum protein signatures of MS patients as well as healthy donors. So these can be depicted like this, which you can see based on the top 10% cellular parameters up to 25 altered serum protein parameters. They were fed into the machine learning tool. The prediction accuracy was quantified by a smooth balanced validated model, and it has been tested again in the in validation cohort. So there are two steps here. First, you get the data, you feed into the model and check whether it is trained properly and it is giving a test result which will classify the samples into three endophenotypes. And when this model is exhibited to the unseen data, it should be able to classify the sample into three endophenotypes. So all patients of these both cohorts were plotted by UMAP using these parameters which I have been mentioning, 29 cellular parameters. And you can see the validation cohort here. The B is the discovery cohort. You can see various colors which will divide all the endophenotypes into E1, E2, and E3. And why should we divide? And these are the graphs which show that there are differences between healthy donors and the endophenotypes, E1, E2, and E3. So this machine learning pipeline was trained initially, as I said earlier, and you can see the training path and the prediction path and the validation cohort. So this model was used to predict the unknown endophenotypes in an independent validation cohort, about 121 healthy donors and 232 patients with early MS. So what it helped to us do us? The prognostic indicators of MS are clinical features, the relapse rate, initial clinical manifestations, early disability accumulation. The paraclinical features are T2 lesion load, GAD enhancement, infratentorial or spinal cord lesions, brain atrophy, soluble biomarkers, etc. These are the oligoclonal bands and chitinase 3-like proteins and the neuronal light chain filaments. So what are the endophenotypes and clinical disease trajectories? The immunological endophenotypes are associated with different clinical disease trajectories. I can summarize by saying that early stages of MS is having these three immunological phenotypes. The E1 phenotype has a degenerative type, early structural damage, and very fast disability progression. E2 is less remarkable, which is because of NK cells, natural killer cells. E3 is inflammatory endophenotype, which has increased signs of early high inflammatory disease activity, which we will describe. The E1 is degenerative, early structural damage, and disease severity at baseline. The EDSS to start with is almost 1.5. The impaired cognition at baseline, music cognition, which is another cognition test, which is less than 20. Increased serum, the light chain neuronal filaments, increased intrathecal IgM synthesis. These are the clinical disease trajectory of this. Whereas E3 got high inflammatory disease activity with higher relapse rate, that is more than 2 relapses within first year, requirement of highly active disease modifying therapies, higher GD, GAD enhanced lesions, higher conversion rate from clinically isolated syndrome to relapsing relative remitting MS, rapid disability accrual within 2 years after the baseline, increased CSF cell count at baseline, increased intrathecal IgG synthesis at baseline. So there are clear-cut differences right from beginning of MS. The data supports existence in early MS with a prognostic significance. So what is this prognostic significance? Distinct endophenotypes in MS lead to 3 different MRI subtypes also: cortical lead subtype, white matter lead subtype, lesion lead subtype. Usually the MRI progression is studied by novel black holes, brain atrophy, appearance of new T2 lesions, enlarging T2 lesions. So these graphs show all these 3 drugs, interferon, gadol, galactomerostate, and dimethyl fumarate are studied about MRI progression and EDSS progression, and they observed some differences, which I'm going to summarize in the next slide. What are the differences that have been observed? See, the differences that are observed very striking, and which the differential impact of distinct DMTs on clinical and paraclinical features of patients with MS with the inflammatory endophenotypes. One is E3, that is the first A and B in this diagram show that is pathophysiology of MS and the effect of disease modifying drugs. The efficacy of these DMTs on parameters associated with MS phenotype was analyzed, and the efficacy of treatment regimes on flow cytometry, also the soluble parameters associated with these endophenotypes were analyzed, and what they observed was very interesting. And the three drugs which are compared, which are used in the early MS, that is interferon beta, glatiramer, and dimethyl fumarate, they used for 1 year and studied what are the differences that are going to happen, that they're going to see. So a very interesting finding has come, and the efficacy is very well studied. And what are the implications? The immune signatures were altered by treatment with DMTs when compared to healthy subjects. So first point. The second point was differences were observed in 38 cellular parameters and 25 serum protein parameters, which normalized with treatment. And there is a difference between the efficacy of treatments. Glatiramer and dimethyl fumarate and interferon beta normalized the immune signatures associated with both E1 and E3, but interferon beta exhibited limited effect on signature alteration associated with inflammation. We have grown with interferons, glatamer, and dimethyl fumarate. Now we know some patients never responded, and this is the inflammatory prototype which I'm going to describe again. So in conclusion, the concept of different endophenotypes in early MS is a result of exploratory research, which is based on a small sample size, and the E1 endophenotype, has shown increased signs of early structural damage, higher clinical disability at baseline. So you can prognosticate, and maybe the highly effective treatment of ocrelizumab, etc., can be started. Whereas E3 is a pro-inflammatory pattern, the increased focal inflammatory CSF alterations, high relapse frequency, and gadolinium enhancement, treatment failures with low efficacy treatments. So we can start with highly efficacious natalizumab and other drugs in these cases. I think that's all from my side about the endophenotypes and current concepts in multiple sclerosis. You all know various therapies that are existing, and these therapies are established by randomized controlled trials, but all of them addressed what is called annualized relapse rate. So when if you want to diagnose MS with immunological basis, we have to study the peripheral blood mononuclear cells, PMCs, subject them to flow cytometry and proteomics, and identify the immune signatures and classify them into E1, E3, and E2, and then prognosticate and then develop the precision model. That's all from my side. Thank you very much. If there are any questions, we can answer, or you can send me my WhatsApp group. Any questions? It's a very difficult concept than what we usually talk. It's basically generated by flow cytometry and proteomic studies, followed by machine learning, model training, and characterization of endophenotypes, which have got a bearing in the pathobiology as well as treatment aspects. So if there are no more questions, then I will say bye-bye to you all till we meet next time. Can somebody unmute your visible? Sir, yeah. Any questions?
No, sir.
Yeah, okay. Thank you very much. Then that's all from my side.